Immunology Insights, Basics and Beyond, Chapter 51, Influenza and Other Viral Infections. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored tuberculosis, a bacterial master of immune evasion that can hide inside our cells for decades. Today, we move from slow and stealthy to fast and furious. Influenza and other viral infections that challenge our immune system's adaptability on a global scale. These viruses mutate rapidly, spread explosively, and test every level of our immune defense, from innate barriers to long-term memory. So let's talk science. Let's start with the basics of influenza infection. Influenza, or simply the flu, is caused by the influenza viruses A and B, members of the Orthomyxoviridae family. They are enveloped RNA viruses with a segmented genome, a design that enables extraordinary flexibility. Each virus particle is covered with two key surface proteins, hemagglutinin, or HA, that allows viral attachment and entry into host cells by binding sialic acid on respiratory epithelium, and neuraminidase, or Na, which helps new viral particles escape from infected cells. Together, these proteins form the basis of the familiar naming system, for example H1N1, H3N2, or H5N1, referring to different HA and Na variants. There are three main influenza types. Influenza A, that infects humans and animals and causes pandemics, for example the H1N1 or the H5N1 avian flu. Then we have influenza B, that circulates only in humans, causing seasonal epidemics, and we have influenza C and D that are generally mild and rare. Some subtypes such as H5N1 or H7N9 are particularly feared because they originate in birds and have high mortality rates, though limited human-to-human transmission. What makes these avian strains so dangerous is their ability to reassort genes between species. When birds, pigs, and humans live in close proximity, such as on farms or live animal markets, influenza viruses from different hosts can infect the same animal, often pigs, and inside these mixing vessels, the viral gene segments from avian, swine, and human strains can combine. The result is a completely new hybrid virus with the virulence of bird flu and the human transmissibility of swine flu, a process called antigenic shift. This genetic reshuffling, often involving the HA and Na segments, can suddenly produce a virus against which the human population has little or no immunity, triggering pandemics. The infection begins when HA binds to receptors in the upper or lower airways. Once internalized, the viral RNA is copied and new viruses are released, often destroying the host cell in the process. Symptoms like fever, cough, muscle pain, and fatigue reflect both viral replication and the immune system's inflammatory response. Well, let's then look at the immune response to influenza in more detail. The immune system mounts a rapid and multi-layered defense. First, innate immunity. Epithelial cells detect viral RNA through pattern recognition receptors like TLR3, RIC1, and MDA5, triggering type 1 interferon production. Interferons like interferon alpha and beta induce then antiviral states in neighboring cells and activate NK cells to destroy infected targets. Second, the adaptive immunity. B cells produce neutralizing antibodies against HA and Na, blocking viral entry and spread. CD8-positive cytotoxic T lymphocytes recognize infected cells via viral peptides on MHC class I and kill them using perforin and granzymes. CD4-positive helper T cells orchestrate this response, providing help to B cells and CD8-positive cells. After infection or vaccination, memory B and T cells remain, enabling a faster and stronger response to future encounters. But why does influenza keep coming back? Influenza's genius lies in its ability to change its appearance faster than our memory can keep up. First, we have antigenic drift, small and gradual mutations in HA or NA that accumulate over time. This is why seasonal flu vaccines must be updated every year. And second, antigenic shift, a major reassortment of gene segments when two influenza strains infect the same host, for example human and avian. The results can be entirely new virus to which no one is immune, triggering pandemics such as the H1N1 in 2009 or the devastating Spanish flu of 1918. Because influenza spreads through respiratory droplets and has a short incubation time, it can sweep across the globe in months, forcing constant public health vigilance. Now, let's look also at other viral infections and immune strategies. While influenza is the classic example of a mutating respiratory virus, similar immunological battles occur also with others. Here are some examples. First, the respiratory syncytial virus or RSV, a major cause of infant bronchiolitis. Immunity is often incomplete, allowing reinfection throughout life. Second, rhinoviruses, which cause the common cold. Over 100 serotypes, making sterilizing immunity nearly impossible. Third, coronaviruses, including SARS-CoV-2. They use spike proteins to enter cells via ACE2 receptors, inducing both mucosal and systemic immune responses. These viruses can cause mild colds or, as we have seen with SARS-CoV-2, global pandemics when novel strains emerge. And fourth, we have the measles virus, which is highly contagious and immune suppressive, and temporarily resets the immune memory. Together, these examples show that our immune system must constantly adapt, not only to recognize diverse viruses, but also to balance response and regulation to prevent tissue damage. Now, vaccination can teach the immunity in advance. Vaccines remain our strongest defense. Seasonal flu vaccines contain inactivated or recombinant viral proteins from the most likely circulating strains, prompting antibody and T-cell memory formation. While traditional flu vaccines offer partial protection, new approaches are on the horizon. For example, first mRNA flu vaccines. Building on COVID-19 technology, they enable faster updates and stronger immune responses. Second, universal influenza vaccines aim to target conserved regions of HA, offering cross-strain protection. And third, intranasal vaccines that stimulate mucosal immunity at the virus entry point, potentially blocking infection before it even begins. These innovations could finally end the annual arms race between viral evolution and vaccine prediction. Let's also have a closer look at the immune pathology, so when the defense actually hurts. Severe influenza infections, especially those caused by avian strains like H5N1, can trigger a cytokine storm, a runaway inflammatory reaction that damages lung tissue and blood vessels and can end fatally. Here, death is often not due to the virus itself, but the immune system's overreaction. High levels of TNF-alpha, IL-6 and interferon gamma cause capillary leakage, edema and acute respiratory distress. In contrast, milder strains tend to kill fewer cells directly and provoke a more controlled response. The virus's danger lies in this delicate balance between replication speed and host inflammation, a theme shared by other viruses like SARS-CoV-2, where severe COVID-19 arises from immune dysregulation rather than the viral load alone. So, what are apt treatments and antiviral strategies? Antiviral drugs such as oseltamivir, or better known as Tamiflu, and zanamivir inhibit neuraminidase, reducing viral spread if given early. Baloxavir, marboxil, a newer antiviral, blocks viral RNA polymerase, halting replication. For severe cases, supportive care and immune modulation using corticosteroids IL-6 blockers, or antivirals in combination can prevent fatal complications. Vaccination and prophylactic antivirals remain the backbone of global prevention. Meanwhile, researchers are developing broad-spectrum antivirals and monoclonal antibodies that neutralize multiple influenza subtypes, offering tools for the next pandemic before it even starts. So, what is its relevance and future directions? Influenza and related viral infections remain a constant test to the immune system's agility. They highlight the importance of first, rapid innate recognition and interferon responses. Second, robust adaptive immunity. And third, global cooperation in surveillance, vaccine development and rapid responses. Future strategies may include mRNA-based multivalent vaccines, universal immunogens, and immune-boosting therapeutics to prevent pandemics before they spread. The COVID-19 pandemic proved that with genomic surveillance and flexible vaccine platforms, humanity can respond faster than ever before. Harnessing the immune system through vaccines, monoclonal antibodies and immune modulation will be central to controlling the next generation of viral threats. And of course, if humanity works together to follow certain safety protocols and rules to prevent further viral spread. To conclude, influenza and other viral infections remind us that immunity is a race against evolution. Each mutation challenges the memory of our immune system and the reach of modern medicine. Yet the same adaptability that viruses exploit is mirrored in our own immune ingenuity. From the seasonal flu to global pandemics, the lessons are clear. Science, vigilance, and immunology must move as fast as the pathogens we face. In our next chapter, we will explore emerging infectious diseases, from COVID-19 to Ebola, and how immunology guides our response to new pathogens that suddenly leap across species and borders. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.